Hello everyone, welcome to today's live broadcast, Improved Decision-Making and Drug Discovery Process of Using an Innovative Approach to Modeling Chronic Liver Disease. I'm Robert Castellanos of Labroots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots. Labroots is a leading scientific social networking website and producer of educational virtual events and webinars, contributing to the advancement of science through content sharing capabilities. Labroots is a powerful advocate in amplifying global networks and communities. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon at the lower right-hand corner of the slide window. If you have any technical problems hearing this presentation, please click on the support button at the top right of the presentation window or submit your problem through the green Q&A button lower left. I would like to introduce today's speaker, Dr. Steve England. Steve is an experienced professional in the early stages of drug discovery and development, from target identification through project initiation up to proof of concept. Steve has a broad-based scientific experience primarily in the neuroscience area and more recently in chronic liver disease and more generally in immunology and oncology. Currently, Steve has two major roles. Firstly, leading the discovery effort on liver disease at AbbVie aimed at identifying treatment for NASH and NAFLD and autoimmune diseases of the liver. Secondly, Steve heads the future of therapeutics and technologies, a team tasked with the identification and interrelation of atypical therapeutic modalities. And now I'll turn it over to Dr. England for his presentation.
Many thanks, Robert. Thanks for that introduction. And before I start, I would like to thank the Labroots folk for the opportunity to present via this webinar. As my title suggests, I actually have dual responsibility at AbbVie. I head Future Therapeutics and Technologies, which is a small group tasked with identifying atypical and potentially disruptive therapeutic modalities. And typically we're interested in things such as cell or gene therapies. And as part of my role, we're also on the lookout for technologies and approaches that more generally add to our drug discovery and development capabilities. So the second part of my role is that I head our discovery group involved in the search for therapeutics for chronic liver disease. And my presentation today combines aspects of both of these responsibilities. I'll talk to you about a new technology and how we apply it to chronic liver disease discovery. So here's the breakdown of my presentation. Firstly, I'm going to start by providing a very personal perspective on some of the factors within discovery that I believe contributes to later stage attrition. I'm going to touch on aspects of systems biology and how I believe the approach and the adoption of the principles of systems biology may address some of the contributors to attrition. I would suggest that the in vivo model probably represents the ultimate systems biology approach, at least within discovery. And I'm going to talk to you about some of the challenges associated with some of the preclinical models in my particular area of interest, which is chronic liver disease. I'm going to present our approach, which actually uses what I believe to be a novel technology, and it focuses on a particular aspect of liver pathology. I'll really go into some detail about how we believe it reduces time, costs, animal use, and maybe more importantly, enables us to make better decisions more rapidly with regards to our programs. So before I start, I'm really going to put out some thought-provoking observations. I believe we need to continually reevaluate how we approach research and development within the pharma industry. And I think it's fair to say we have to look ourselves in the mirror and have to agree that across the industry we probably haven't been as productive as we need to be. So why is this? Well, I've just bullet pointed here some of the reasons that I believe could be contributing factors. Again, a very personal perspective, and these are meant to provoke thought. But I think as technology advances, we're increasingly aware that disease biology is extremely complex. And we know if we aim at a single novel target, very often they don't translate into clinical success. And I also think it's fair to say that there might be one or two exceptions to this, but I think out of infectious disease, there are actually few drugs that alter the underlying cause of disease. Most of the treatments that we currently have are symptomatic in nature. And I still think there's a large amount of potential target space that we regard as undruggable. And by that I mean we view them as undruggable because we think small molecule and large molecule approaches. And I think this is likely to change in the future. So although we have an increase in the knowledge of the basic science, I actually think that isn't really translating through to innovation in how we do our drug discovery and how we do development. We know the cost of drug discovery and development is increasing, and maybe the checkpoint inhibitors might be the exception to this, but I think there are very few new game-changing therapeutic modalities. And I think very interestingly, medicine isn't different from that practice decades, maybe even hundreds of years ago. It relies on an individual becoming ill and then going to their physician who would prescribe some kind of medication. But I think as we understand more about the genome, the whole emphasis of how we treat disease is going to shift to prevention rather than cure. And I think that is something that Big Pharma really will have to get to grips with in the not too distant future. And having said that, I still think Big Pharma's approach to drug discovery has been largely unchanged over the years. So this is the usual schematic of drug discovery. I do not like this approach because it makes a number of assumptions. Firstly, it assumes that we have a fixed rate of attrition, such that in the discovery phase, we tend to see the duration of getting a preclinical asset into the clinic of somewhere between 6 to 8 years. It suggests also, I think, that we are locked in to research and development timelines. If we were to map on top of this continuum, there would be a large inflection point when an asset crosses from the preclinical phase into the clinical domain. And I think what that means is, for us, very often we think that the greatest economies can be brought within clinical development. I actually think that making better decisions within discovery will help reduce our attrition rates. And I think anything that moves us away from the shots on goal philosophy, such that all we need to do is increase the number of assets to the left hand of this continuum, that's all we need to do to increase our success in terms of marketed products. I think this is really something we need to challenge most vociferously. So I think one of the most eloquent commentators on this subject is Bernard Munoz, and here are a couple of snapshots from, this was a commentary piece that came out last December. I would encourage anyone involved in pharma R&D to seek out and read his work. Put simply, this says that the industry has been victim to a perfect storm of circumstances. The molecular biology revolution enabled us to identify, to clone, to express potential targets of choice, and it became extremely easy to make cell lines and run things like high throughput screens and to trigger drug discovery programs. It was very easy to be reductionist. This was coupled to enhancements in medicinal chemistry, for example, combinatorial chemistry. So overall, what this meant was that pharma companies could generate huge compound libraries running into the many millions, coupled with consultancy firms telling us all that really what it was all about was shots on goal. And we just needed to increase our productivity at the discovery side of the business, and we would have increased success within the clinic. I think this is actually very wrong, and I think we need to challenge this very firmly. So how do we think about this? Well, maybe as a backlash to the reductionist approach emerged the, I think, increasing understanding that actually moving away from the reductionist approach and more into a systems biology arena would provide more success. And this, if you like, spawned the systems biology motion. And this really speaks to the importance in having a greater understanding of the interaction of players within a biological system. And this was championed by Leroy Hood. And I would argue that one of the reasons for attrition was that we've been too keen to follow the bystander players as shown here. If we consider these are molecular pathways, and I think we've been too keen to look at the bystander players in this, as shown by the red arrow, and I think we've been less interested or less able to find what we regard here as choke points in the system to enable us to have a better chance of getting a bigger bang for our buck when we go into the clinic. So I believe systems biology is important, and I think it can derive a different way of thinking, and this is really summarized on the right-hand side. And what this actually means is that as technology advances, we generate massive data sets, and this will enable us to be much better at understanding very complex biology. This in turn will generate new questions to which we need to go and find answers. And so this is really how our understanding is likely to advance in the future. It will move hand in hand with technological advances. So, okay, this might seem all well and good, but how do we actually apply these principles to aspects of the drug discovery program? So here you can see the traditional preclinical drug discovery continuum. Right from the left-hand side, we identify targets, we generate screens, we identify hits, we exemplify those hits using medicinal chemistry, and eventually we move these into in vivo models. It's the right place to have the in vivo models on the right-hand side of this, but I think we can be much better in applying the principles of systems biology to in vivo modeling, and also keeping to the tenets of 3 R's. And I think this is really important. We need to avoid using animals as screening tools, and if we can make more effective decisions earlier on, reduce the number of animal usage and reduce the number of model studies that we are using, then that has to be good for the 3 R's. So I'm now going to change tack somewhat and tell you a little bit about my area of interest, and this is chronic liver disease. So chronic liver disease is a huge public health problem. Greater than 1/3 of the US citizens will have some degree of chronic liver disease, and thankfully for most, this will be mostly benign fatty liver disease or steatosis. However, for some individuals, the condition is severe. It can lead to inflammation, the central point here, and once inflammation is triggered in the liver, it can cause increasing levels of fibrosis, ultimately leading to cirrhosis. Now, I think most of us will be more familiar with alcohol-induced cirrhosis, and that's reckoned to be a fairly severe disease, but this is a much more severe healthcare time bomb, as this non-alcoholic steatohepatitis is driven primarily by poor diet, obesity, and diabetes. So as the incidence of diabetes increases in the US population, we would expect to see more patients suffering from chronic liver disease. So as liver disease progresses, there are increased levels of fat laid down in the liver. And under normal circumstances, the liver is actually designed to handle lipids. But if the amount of lipid overwhelms the ability of the liver to deal with it, it can trigger inflammation, shown in the central portion of this cartoon. This leads to NASH, non-alcoholic steatohepatitis, and this in turn begins the process of the liver laying down increasing amounts of collagen and other structural proteins that make up the extracellular matrix. Once the liver becomes more fibrotic, this is much more serious. It can result in the loss of metabolic function. The liver loses compliance, and by that we mean it loses its spongy texture and becomes much more stiff. Maybe more importantly are the cardiovascular consequences of an increasingly fibrotic liver. These are extremely severe. They lead to portal hypertension, esophageal varices that can bleed, encephalopathy, and eventually death unless the patient receives a transplant. So this is a very big deal, and many pharma companies are engaged in the search for effective treatments for aspects of chronic liver disease. Now, AbbVie is no different in this. We have an interest in this area, but I'd like to say a little bit about how we're approaching this, because I think it's a little different from the norm. The field is moving extremely fast, and we have to be competitive within this space. So we have a time-bound engagement, and by this I mean we do not have an open-ended commitment from our senior leadership here. We know we have to enter the area, succeed in the area in a very short order. But really, to succeed in this mode of working, we have to think differently, and we certainly have to adopt a very different operating model to the traditional pharma way of working. A traditional pharma way of working would be to go out to hire the relevant experts, to initiate target identification, to make cell lines and initiate high-throughput screens. And all of this constitutes a very long time lag, and of course none of that ensures success. So we've got a slightly different way of approaching this. Now, this also creates a problem when it comes to preclinical models of liver fibrosis, because these are chronic. It depends variably on the stimulus, but usually these run about 6 weeks. And so if we consider the chronic nature of the in vivo models, there is also a certain degree of fuzziness associated with the endpoints, because these are very usually histology. And so overall, the in-life component and the post-study analysis is extremely labor-intensive and very time-consuming. So what do I mean by time-consuming? This is a screenshot from a review that highlights some of the preclinical models of fibrosis. Things to take note here are the durations for fibrosis induction in these models. And as you can see, the time is variable, but the time needed for pathology to develop is dependent upon the stimulus, but is usually pretty long. Not included on this slide are some models of liver fibrosis due to diabetes and diet-induced liver damage, and those models can actually last upwards of 6 months to enable the fibrosis to develop in the liver. So if you consider running these chronic models with compounds of interest isn't really in keeping with our desire to be very agile and flexible. So what we did was decided to adopt a slightly different approach and focus on the inflammatory component of liver fibrosis. And that's relevant because, as I mentioned before, it is the driver of fibrosis, somewhat independent of the model that we use. So we actually set about modeling the inflammatory component of liver disease using concanavalin A. So a couple of things that are really important to point out here. We're no longer focusing on the anti-fibrotic mechanisms, just the upstream driver. So this is only relevant for agents that are inhibitory to some of the inflammatory processes. We actually think this is justified, provided we use compounds which we think work via this component of the pathology. Now, to do this, we use concanavalin A. conA is a plant lectin. It has been widely used to induce hepatitis in mice, and also explore some of the pathophysiological inflammatory mechanisms downstream of conA. Now, this has a relevance because many of the pathogenic drivers in human immune-mediated hepatitis, so by that we can mean autoimmune hepatitis, certainly viral hepatitis, and maybe even some drug toxicities, seem to use common immune pathways and inflammatory mediators. So we believe this is valid. So conA produces a biphasic effect in the liver. I'm going to talk about this a little more in a moment. We have a direct cytotoxic effect on hepatocytes, and this also causes a significant infiltration of T cells and the release of large amounts of inflammatory mediators. And again, the importance of this is that inflammation is the driver for laying down the extracellular matrix in the liver and the initiation of the fibrosis. It's also worth pointing out that the single injection of conA, at least in the mouse, actually produces a very acute inflammation in the liver, and pretty much the inflammation in the liver is finished after a single injection after about 24 hours, after which the liver returns to pretty normal function. So this is one of our internal experiments. This pretty much shows histologically what I'm speaking to. So on the left-hand side, we see a histological section of liver from a mouse treated with vehicle, and here the red and the black have been used to stain inflammatory cells in the liver. The middle panel type in conA shows a mouse who has received a single injection of conA, and you can see there's a high density of both red and black stained cells in the conA liver. And on the right-hand side, this is a separate experiment, again a histological sample from a liver from a mouse that had received conA, but in this case, prior to conA, this mouse had received dexamethasone. Dexamethasone is a steroid which suppresses inflammation, and as you can see, the right-hand panel shows a significant decrease in the levels of red and black stained cells, which again is consistent with the anti-inflammatory properties of dexamethasone. So it's important to show a little more detail the mechanism of action of concanavalin A, and this cartoon shows the mechanism in a little more detail. conA actually binds to a mannose receptor on the sinusoidal endothelial cells, labeled SEC here. And this is in the liver, and what this causes is an acute breakdown in the tissue architecture. Now what this does is exposes the Kupffer cells, KC in this diagram, which are the resident macrophages in the liver. Secondary to this, part of this biphasic effect, is a T cell infiltration, and this T cell infiltration really underpins the acute inflammatory response that we see to conA. As the immune system really kicks fully into gear, we actually see a whole range of cytokines and chemokines released both into the systemic circulation and also in the liver itself. And the right-hand panel here shows, in the top part A, shows systemic release of a whole range of pro-inflammatory mediators, and underneath we can see the liver levels of the same inflammatory mediators. And so this illustrates a couple of things. Firstly, illustrates the acute nature of the challenge, because if you look at the 24-hour time point here, pretty much the cytokine and the immune modulator release is pretty much over and done with, both in the circulation and also in the liver. So we actually think this has a clinical relevance, and so we think it has a clinical relevance, again, because many of the processes that we stimulate using the conA model are also seen in human diseases such as autoimmune hepatitis, primary biliary cirrhosis, and primary sclerosing cholangitis. And in actual fact, we can also add to this the inflammation caused by metabolic drivers, the obesity and diabetes induced inflammation in the liver. So we think that there is a real relevance to conA in modelling the inflammatory drivers in chronic liver disease in man. So, to summarise our thinking, where we are up to now. So, we know we can evoke an acute inflammation in the livers of mice using concanavalin A. We know that we can focus on the inflammatory drivers of liver fibrosis. By doing this, it may enable us to triage compounds of interest more rapidly. And it doesn't mean that we are replacing more chronic models. It means we have an ability to triage compounds through the conA model early, and so this means that we can go into more chronic models with a fewer number of compounds, each of which will have a high confidence and probability of success. Having said that, even if we assume the conA as the model and move to this more acute model, it still involves a histological evaluation of the liver or a biochemical analysis of the liver after the study has been done. Also, it's quite involved if you want to look at inflammatory mediators in the circulation. So what this meant was we actually came to think that maybe there might be a better way of doing this. Now, what we know is in humans, when we evoke large amounts of systemic release of pro-inflammatory mediators, it produces what is known as a cytokine storm. So this cytokine storm produces a fever and a general lethargy and fatigue in patients. So we hypothesized that there might be an analogous behavior that we might be able to measure in mice. And so collectively, this thinking took us towards the Vium Digital Vivarium technology, which I'm going to talk about in a moment, because we believe that maybe we could identify a behavioral endpoint against which we could test compounds of interest. So what I need to do is provide you with some background to the technology, and it should be important to point out that at this stage I actually have no financial linkage to the company whatsoever. It's just an excellent technology, and we've applied it, I think, in a very novel way. So Vium has their digital vivarium, and what this offers is the ability to continually monitor mouse behavior. Now, this is done in a remote fashion and with minimal human intervention. Now, the way this works is that the individual cages are wired with all sorts of sensors to monitor both environmental conditions and also a multi-parameter assessment of mouse behavior. So what this actually means is that you can assess in real time the activity of the mice within the cages, and this is done because there are cameras within the cages, and this means that the mice are monitored 24 hours a day continually. So this is a fantastic technology, but maybe more importantly, the technology is sensitive enough to measure respiration rate in mice. The technology does this by detecting the movement of the mouse chest area during periods when the mice is stationary. And I think this is really important because this is one of the significant advantages that the technology offers, because there is continual assessment of animal behavior, I think, in a way that would be impossible by a human experimentalist. And it would be interesting to meet anyone who could eyeball the mouse respiration rate over a period of 24 hours. And this is something that the Vium technology offers us. So when you run these experiments, what do you see? So here we have on the top panel measurements of activity in a mouse relative to circadian rhythm. So this is a single mouse in one of those activity cages. The greyed out area shows the dark cycle, so there's a 12-hour dark cycle, and the white intervening bars show the light cycle. Now, what we actually have here on the left-hand panel of the upper data here, you can see that on night 1, as the dark cycle starts, the animal becomes very active. And over the course of the 24-hour period, the animal is active, but it gradually tails off over that 12-hour period. And as you can see in the light cycle, there is almost no activity of the mouse, and this is maybe not surprising because the mouse sleeps during the day. So what we actually can see is on night -1, this is the mouse doing his natural thing in the cage. At night 0, concanavalin A has been applied midway through the dark cycle at the black arrow here, and as is clear here, there's quite a rapid decrease in the amount of activity in the mouse. As we might imagine, the following light cycle, the mouse is pretty quiescent, but what we can see on the second night is that there is a significant suppression in the activity of the mouse in response to conA. The bottom panel here shows, this is collated data from groups of 6 mice and a dose-response curve to concanavalin A. So there are a number of things that we can take from this data. It's interesting that with 0 conA, so this is a non-inflamed liver, there was less activity on night 3 relative to the activity that was seen on night 1. So night 1 being the white bar, night 3 being the black bar. And I suspect what this reflects is less exploratory behavior in a mouse that was a control, and this reduction in exploratory activity probably reflects the animal becoming more comfortable in the cage and probably requiring less exploratory behavior. So we can see this in the controls. 15 milligram per kilogram conA actually didn't evoke any change in behavior, neither did 20 mg per kg conA, but as you can see at the top dose here, 25 milligram per kilogram conA, there was a significant reduction in the activity of the mouse on the night after injection. So really what we have here is this is actually in keeping with our original hypothesis and belief that circulating cytokines would produce a state of relative inactivity in the mice, and this could be analogous to the lethargy fatigue that is seen in humans in response to high levels of circulating pro-inflammatory mediators. So this was incredibly encouraging for us. So the next slide shows that in actual fact conA was actually having the effect that we thought it would have. So this is not behavior, this is measurement of circulating liver enzymes. ALT and AST are enzymes that are actually used in clinical practice to assess the state of liver health. And if you look at the data in both the left and right panels, it's clear that the top 2 doses of concanavalin A produce significant elevations of circulating liver enzymes, and this is again consistent with our hypothesis. The 25 mg per kg dose produced a substantial increase in both ALT and AST, and this is consistent with what we know to be happening, the inflammation that is occurring within the liver. Now there is an interesting aspect to this, and although the 20 mg per kg dose of conA actually caused an increase in AST and ALT levels, if you recall from the previous slide, only the 25 mg per kg dose suppressed activity. So there seems to be some separation here, and one potential interpretation of this is that the higher dose probably evoked a greater degree of mediator release, and that was what was necessary to actually evoke the change in behavior within the mice, despite the fact that there was still some ongoing inflammation in the liver. So the next slide shows where the Vium technology really provides a significant advantage over more conventional approaches. So here we can see the change in respiration rate in a number of mice in response to increasing doses of concanavalin A. Now clearly there is some overlap in the data here, but in general you can see that there is a dose-dependent increase in the respiratory rate in response to the conA challenge. Now, really, if you think about this, the Vium technology here is measuring respiration rate by monitoring the movement of the chest wall of the mouse when it's actually static over a fixed period of time. If you actually stop and think about that, that is a pretty phenomenal ability of the technology to pick up this kind of change. Now, it's still very early days, but we actually believe that we can actually use this to screen compounds that might suppress inflammation in the liver, such that we can use this rather than have post-life analysis to determine what compounds we want to advance into the more chronic models. So this is how we see the approach. We can rapidly assess compounds using the behavioral endpoints. We can actually see in real time whether our compounds are having any effects. We think the study execution is going to be pretty rapid. We can follow up with post-study analysis, the biochemical analysis of both tissue and also plasma. But we don't have to rely on these to make decisions because of the real-time nature of the behavioral endpoint. So we think we'll be able to make decisions on compounds rapidly, decide what we want to take forward into the more traditional models of fibrosis. And this slide really shows the value proposition that Vium provides to us. And I think, importantly, what this means is that we have the ability to use less compound, we can use less animals, the severity of the models that we use is lower. So this is pretty much in keeping with everything that we would hope for from the point of view of aspiring to best practice in 3 Rs. I think another hidden cost to running chronic studies that we've not mentioned now is the amount of compound that we actually need. If you imagine the amount of compound that you would need to support a 6-month diabetes model involving daily dosing, then that is a significant compound investment. And so what we think we can do is use the Vium technology to measure behavior. We can pre-dose with a single dose of compound, inhibit this behavioral endpoint as a surrogate measure of compounds that actually suppress the inflammation. So, how do we see the future? Well, we hope to be looking at this particular behavioral endpoint in a little bit more detail to work through more and more pharmacology to really understand more about what the model is telling us. But what we also hope to do is work together with Vium to explore mice with humanized immune systems. So this is just screenshots from three papers relating to this. It would be fantastic if we could work with mice with humanized immune system, as we believe this will give us more confidence about clinical translation. Here are just some examples, but there are many more, and I think this is an area that we're going to see advancing in the future. So if we consider the notion of humanized mice, we are beginning to see in the literature examples of mice with humanized liver. So if we take this further, we would like to explore this acute inflammation model in mice that have both humanized liver and immune systems. And we begin to see this in the literature, and I think this is a field that will advance very rapidly. And we believe that the humanized liver, humanized immune system mouse could provide us with an improved forward translation of assets as we move them into the clinic. So we also do some post-study analysis as well. But in keeping with our nimble and agile approach, I want to tell you a little bit about how we operate with another company called Transcriptic. Transcriptic works through a fair amount of our post-study analysis. So Transcriptic are cloud-based. They have a very flexible lab-in-a-box system which enables you to plug and play depending upon assays that you want to run at any given point in time. And again, this is something that you can run real-time. You can see via the cloud your experiments being run and all the quality control that goes with it. And in actual fact, Transcriptic are performing much of the analysis of the blood samples and also assessment of PCR from the liver in our models as well. So not only do we believe we have a very efficient way of looking at the in-life component of this particular model, but we're also adopting a much more flexible and rapid approach to post-study analysis. So if we consider how we're actually working on this, we will continue to do maybe conventional approaches to identify hits. We believe that the Vium behavioral model could enable us to have a high throughput in vivo engine through which we can rapidly profile compounds of interest. As I mentioned before, in the future we hope to be profiling our compounds in humanized mice, given the increased relevance that we believe this holds for us. We still propose to do the chronic studies with, for example, carbon tetrachloride, and we may even be able to do these models in humanized mice. And by working through Transcriptic, we get a very rapid turnaround in the data. So if we consider how we are working, so this is the drug discovery paradigm, the discovery paradigm that I mentioned before. I think we're actually approaching this in a different way. We're actually building out our capabilities using the Vium technology for fast-track in vivo profiling of compounds, and we're using Transcriptic to enable the bioanalysis to really accelerate our drug discovery programs. And we believe this is a very flexible way of working. It increases our virtual footprint working through high-tech external companies. So, to bring all this together and to summarize, we actually believe that discovery has a vital part to play in reducing attrition in the drug discovery development continuum. We think that adoptions of systems biology thinking and a move away maybe from the over-reductionist approach will be key to reducing attrition. But when we think about this in the context of in vivo modeling, it does produce challenges in keeping with best 3Rs practice. But I believe that by being smart and adopting some lateral thinking, we can actually embrace systems biology in our in vivo models. And I think, moreover, it enables us to improve our decision-making process. We can do this in a way that is completely in keeping with good 3Rs practice. And with that, I'd like to thank you for listening.
Well, thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question for you, Doctor, is this. How widely do you think the mouse automated activity tracking can be applied beyond your example of acute liver inflammation?
I think that's a really good question. I think the answer to that is very widely. I think there are clear applications in the neuroscience area. There are numerous models of neurological disease, and many of these are based around rodent activity. I guess one of those would be, for example, maze paradigms. That would be a reasonable example. And I think clearly the automated nature of the technology would be really advantageous. If you consider learning and memory, it's I think reasonable to assume that you could introduce some learning and memory paradigm into the activity within the activity cages, and I think this will give you a very hands-off and very data-driven ability to measure the pharmacological effects of compounds. And I think we know that there is operator bias that significantly impacts the ability of our models to generate sound and robust data, and I think automated hands-off methodology will certainly help with that. One area where I think there is clear benefit from the digital vivarium is in analgesia, and I think the analgesia field pretty much has been held back largely due to the shortcomings of the in vivo models, and I think clearly the Vium technology has a direct relevance there. I think one of the important aspects in the future is that payers, not only will we need to show significant benefit with regards to patients' disease, but I also think we would need to show improvements in their quality of life. And I think if we then consider what quality of life might look like in the preclinical models, then I think this is something that the Vium technology could be applied to, because we know in many human diseases there are changes in activity. Disruption of sleep is very common in patients with a number of diseases, and I think maybe the Vium technology may be able to begin to model some of these, if you like, more fluffy endpoints involved in quality of life. And I think clearly a direct application of the technology will be in certainly lung research. The ability to measure respiration rate directly can be applied to models, I guess, of COPD and lung fibrosis, and I think this is a very significant advantage of the Vium technology. I think more broadly as well, clearly their applicability into side effect testing of compounds of interest. So I think it has massive potential.
Well, thank you, Doctor. And it looks like we have time for one last question here. Can you provide a little bit more context on how this acute model will affect your decision-making process regarding the use of more chronic models of liver fibrosis?
Yeah, I think that's a really important point. And what we are not suggesting is that you are not going to perform the chronic models. What this really does, we believe, it enables us to have much more confidence in those compounds that we actually take forward into these more complex models. So if we were to go external with a chronic liver study, if we were to go to a CRO, that CRO is likely to charge maybe upwards of $120,000 for a drug study in, for example, a diet model. In actual fact, it could be more if you want to include maybe harvesting a tissue and plasma sampling. And so, it's clear that with that cost and maybe the time associated with running these studies, it's clear that you need to be pretty confident in what you actually progress into these more chronic models. It's not an area where you want to go fishing for pharmacology. So we believe the acute inflammation model enables us to make more decisions on more compounds, and then what this actually means is you end up taking fewer compounds into these more expensive, more time-consuming studies. And I think also, if you actually also perform these studies internally, there is still a tremendous amount of dollar cost involved in this. You have sunk costs, and you also have the invisible costs, if you like, associated with running these studies. So I think it's not an either or. This is all about adding to the screening funnel, and it's primarily about aiding better decision making within our drug discovery processes, and ultimately reducing animal usage completely in keeping with aspirations of good 3Rs practice.
Well, Doctor, before we wrap up today, do you have any final comments?
No, again, I'd like to thank the organisers for inviting me to speak. I'd like to thank everyone for listening, and feel free to email me with questions if you would like any follow-up.
Well, thank you, Doctor, for your time and your wonderful presentation, and I'd also like to thank Labroots for making today's webcast possible. Today's webcast will be available for on-demand viewing through April 2017. You'll receive an email from Labroots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event, and until next time, goodbye.